The class after the last grade game. That's crazy. My grade actually went up to the A for the first time.
Hey, I wanted to actually propose maybe a little change in our test schedule here. So anyway, we are going to do lecture exam 3 in 2 weeks, right? That was going to be before Thanksgiving break, so you all can just relax over Thanksgiving break and not remember anything. It's great. Anyway, so that's lecture exam 3 for sure. And then I had said that we were going to do a lab exam and lecture exam both this week. I'm thinking that's a lot for you, because they probably have other tests. How about we do this? How about we do the lab exam on December 12th? That's lab exam 3. That's the last lab exam. That's the week before. They don't call it finals week anymore, but it's called finals week. So anyway, we'll do the lab exam December 12th. And then that way I can get that back to you on the 17th, and we'll be able to do a review for the lecture exam, right? And then we'll do the lecture exam. Does that sound better? Rather than exams along with all your other exams. And maybe on the 17th, I can ask a favor. I might even provide some extra credit. So let's give it some extra credit already. We'll do a lab cleanup on the 17th, maybe. And you can help me just put the stuff away and organizing whatever. Not too much. I appreciate that. And I'll provide some quick encouragement.
Did you say the December dates again? Sorry.
December 12th is the lab exam.
Okay.
So Thursday, I like to have exams on Thursdays because
I'm sure hoping my oceanography field trip is this day and not this day.
Okay, anyway, and also because then we have an opportunity to review just 2 days before. Whereas I find when you come back from the weekend, like you got about 4 other classes, other stuff going on. I have empathy. All right. I guess it's long time ago. I know. So anyway, lab exam December 12th. That's our lab exam 3. That's our last lab exam. And none of these are cumulative. These are all just the information leading up to that test, right? And then the lecture exam 4 on the 19th. Got it? Okay, cool. I thought about that the other day after I told you. I'm like, man, two tests. So, okay, let's go ahead and get on with our with what we're doing today. We're going to finish today the arthropod physiology. So this is like last time we talked about respiration in arthropods and how 3 groups of arthropods potentially colonize land, right? That's a really important idea because considering arthropods were the first animals to colonize land, they probably did it like several times. Let's see, book gills to book lungs is one event. Gills to using a bubble to cover your gills is another event. And then lastly, how the insects develop tracheal system, which is a totally unique to terrestrial environment type situation. So I think that's really important question, as well as the whole idea of why live on land, other than having a salad bar that no one else was using, right? Because that makes sense because there's food resources when plants colonize land. The other interesting thing is that there's so much more oxygen available. I think it's like 32 times more oxygen concentration per volume, and the ratio of oxygen to CO2 is staggeringly different. You know, in water it's like 2-to-1 ratio, and on land it's like a 500-to-1 ratio. So what we'll see when we talk about respiration in vertebrates is organisms like catfish and goldfish, if water gets too warm or lacks oxygen, they'll go to the surface and grab gulps of air. So even aquatic organisms will go suck up atmospheric oxygen to try to make it through low oxygen times. All right, let me get my my thingy here. Okay, and then today for lab, we're going to jump into our last group of vertebrates here. Okay, invertebrate physiology 3. Okay, cool. Exciting stuff. All right, so let's go ahead and cover osmoregulation in arthropods. And the one theme that I would like you to know or remember, and certainly as will appear on an exam, is arthropods, like nematodes, don't have any ciliated cells, right? There's no cilia. It's unclear why this is. You'd think that ciliated cells would be like a basic thing. Paramecium are ciliated organisms, Ciliophora. Ciliated cells occur in virtually all the other invertebrates. But for some reason, nematodes and arthropods don't have ciliated cells. It's weird. So anyway, it might be because of how they do their molting. They shed their exoskeleton to grow. Nematodes do that, and arthropods do that, but no one really knows the answer why they don't have ciliated cells. But that being said, without ciliated cells, you can't have a nephridia, right? Nephridia, the definition of nephridia, which occurred in the annelids and the mollusks as their osmoregulation and nitrogenous waste balancing organ. You can't have a nephridia if you don't have cilia. So I think this is important because they have to come up with some alternative. So let's go ahead and take a look at crustacean. Black. So crustaceans, they're all osmoregulators, including the ones that are in marine environments. So you kind of get now, hopefully, that osmoconforming is an easy, kind of a lazy, sort of a lazy way to deal with your environment. But the problem with osmoconformers is you're limited to just living in the ocean, right? Like we'll talk about that with the echinoderms today. The starfish, sea stars, and sea urchins, etc. Very diverse group. They've basically been around since the Cambrian, very successful, but they only live in the ocean, and that's because they're osmoconformers. So being an osmoregulator gives you a lot more options for where you can live, not only in the marine environments, but moving into freshwater or terrestrial. So when we're looking at crustaceans, look at, say, for example, our something like our let's see here. Gotta draw a crayfish or something like this. Gotta have 10 legs. Okay, so here is our crustacean, like our lobster or our crayfish or crab or shrimp or whatever. And I mentioned that they have this kind of a unique organ that is called a green gland. And the green gland, some of you saw this when we were dissecting the crayfish. Mostly if you kind of completely pull the head off, you can find it. But the green gland is a osmoregulatory organ. It's not doing any nitrogenous waste balance because these crayfish, for example, or whatever, have gills. The gills that are branched off the legs, they can simply get rid of ammonia through the respiratory surface. So ammonia or urea or whatever they need, usually it's just ammonia. They're able to get the ammonia basically out very quickly because you saw all those gills, that big old fluffy kind of pom-pom-like gill structure on them. Getting rid of ammonia is relatively straightforward. Even our terrestrial crustacean, little pill bug, excretes ammonia out of its water bubble and its gills. So they use simple diffusion from the gills to get rid of ammonia. So that being said, the green gland is not really a nitrogenous waste excreting organ. It's purely a water balancing structure. So anyway, the green gland uses osmotic pumps. What it does, it uses salt pumps to try to excrete water. So in say, for example, a freshwater crayfish, let's use a freshwater crayfish as an example. Crayfish or crawdad. They will collect water out of their hemocoel, transfer it into the green gland, and they do this, there's no like a pump that can pump water, but what they do is they collect salt ions, salts, sodiums and chlorides, etc., and they'll pump those into the green gland. That makes the green gland hypertonic, so it sucks water out of the hemocoel, and then water gets pushed out of their antennal gland, the little gland facing the antenna. And it's sometimes fun. We don't have any live crayfish here, but if you pick up a crayfish out of the water, oftentimes they'll be spraying water out of their face, and some people think it's like them peeing on you, but they're not actually. It's not urine. It's basically just water being pushed out of their antennal glands. So I think the main thing I want you to know or remember, and I'll probably have this as a important point on the exam, is the green gland has no cilia. And so they have to come up with some alternative organ, and they've developed this off the base of their second antennae. I would say, if I were to compare this, like where did this come from? This is probably very, very similar to the excretory duct or the pseudonephridia that we saw in the nematodes, right? The nematodes had, I think it was a pair of excretory ducts that ran down the side of the body and then exited near their head. And they functioned very similarly. They used osmotic salt pumps in order to draw water out of the body and push water out of the organism, or as needed. And the nematodes also had no cilia. So if I were to say, what's the green gland? Maybe it is a pseudonephridia. And I made that term up. But you should probably think of it being like, this is probably very similar to what the nematodes use for osmoregulation, okay? This little chart here is kind of cool. I mean, you don't need to worry about it. But what this is a way to determine if an organism is a osmoconformer or an osmoregulator. So this x-axis is the percent saline in the external environment. Seawater is at about 3.5% saline. And this is the percent saline in their internal hemocoel. And if it's a 1-to-1 ratio, if you're an osmoconformer, you would basically be parallel with that line, right? So you can see that this shrimp species, Krengon, is able to actually maintain a higher internal saline at low, like in freshwater environments or maybe in brackish water environments. And then in hypersaline environments, like that are 4%, think of like the Salton Sea or the Great Salt Lake in Utah, they can maintain a lower concentration. So the flatter that line, the better the osmoregulator they are. So, I mean, that's just kind of some data showing you what osmoregulation is. And it means that they can actually osmoregulate in freshwater and in hypersaline water. So it's pretty remarkable. Okay, key thing though, and what I really want you to make sure you know on the test, so say this on the test. Green gland is a unique organ here because they have no cilia. They can't have nephridia. And so they're using basically a system that's probably very similar to the nematodes, which is this osmotic pump structure in order to bail water out or retain water in those environments. Okay? All right. So when arthropods move onto land, the big issue obviously is you can't use really like osmotic pumps directly to just kind of move water in and out because water loss is an issue. Also, if you move onto land, you can't excrete ammonia. It's really just too much water loss to excrete ammonia. So the terrestrial arthropods, namely the insects, scorpions, spiders, millipedes, and centipedes, they have developed a totally unique, different way to excrete nitrogenous waste, which is pretty remarkable. And this is what's called the Malpighian tubules. You know, I was all gaga about Malpighian tubules, the insect, the cockroach. So let's go ahead and talk about how the Malpighian tubules work. So this will be the insects. It also occurs in scorpions, spiders, millipedes, centipedes, and it might have actually independently evolved in all these groups. Because I've already kind of implied that scorpions probably colonize land early, and insects maybe colonize land later, that when each of these groups colonize land, they kind of independently evolve in a similar type of system. But anyway, let's go ahead and draw a basic insect here. Here's our insect. Eyes. All right. So let's see here. We have a crop. We have a pharynx. Crop CE. Midgut. Okay, and then the hindgut, or the colon, is here. Here is the hindgut. I guess you could call it a colon, but it's usually called a hindgut in entomology, anatomy and stuff. And then, at the junction between the midgut, which is the absorptive part, and the hindgut, so the midgut absorbs nutrients, and the hindgut resorbs water before the feces are excreted. There are these structures, which are, let me go ahead and, I'll do this. Try to think here. These are and the name Malpighian is there's an Italian biologist back like in the 1800s named Malpighian that described it. So, you know, that's the great thing about finding things you can name after yourself, right? I'll give his name later, but anyway, the Malpighian tubule is kind of an interesting spelling, but it's named after a person. There's no functional name for it. So anyway, the main thing to note is these are these tubes, closed-end tubes that branch off the junction between the midgut and the hindgut. And what they do is they kind of lay in the hemocoel. These kind of remind me of if you've ever seen someone's swimming pool that has those hoses in the bottom that kind of snake around. And the hoses that snake around, I think the idea is they're supposed to like wash algae off the bottom of the pool and do the filtering process. But anyway, they work the same way. They actually kind of snake around the inside and they move. When the cockroaches are fresh, you'll actually see the Malpighian tubules kind of moving. There's a little musculature in them, they kind of wave around. But they're collecting water salts and nitrogenous wastes from the hemocoel. So let me go ahead and draw kind of a large diagram of what's going on here. And so we have our midgut. And again, this is probably something like you're going to have to do on the test. Okay, so here's our hindgut. Here's our midgut. All right. And then we have our Malpighian. I'll just draw a couple of them, not all of them. But we have our Malpighian tubules, which are these. branches. And again, there's no cilia, so this is like they have to come up with some alternative way to do nitrogenous waste balance. All right, so what they do is a couple key important things here is that the pH or the acidity of the hemocoel is, as you'd expect, a pH of 7. So the pH or the acidity of the hemocoel, let me go ahead and do this, let's see here. Yeah, that's fine. The pH of the hemocoel, the capillaries, is about pH 7, which is neutral. This is a, because it's the body fluid, the hemolymph blood fluid needs to have kind of a near neutral pH in order for most enzymes and cells to function properly. The pH of the hindgut is usually fairly acidic. The pH of the hindgut is roughly 5-ish, 5 to 6 or so, depending on where they live. So that's kind of like a key mechanism here for how this works, is that in the hemolymph at pH 7, you have your hemolymph fluid circulating around. Remember, in the insects, the hemolymph doesn't carry any oxygen. There's no blood cells, there's no hemocyanin or any kind of blood pigment in here to carry oxygen. The oxygen is carried to the cells by the trachea. So the hemolymph here is just for carrying around nutrients and waste. So anyway, in a pH 7 environment, we have in this hemocoel, we have a couple different things going on here. Let me go ahead and see if I can use some so we have water. Water. So we have water going on in here, pretty obviously. And then we have nitrogenous waste, which is uric acid. So water, uric acid, and let's go ahead and add one more thing, which is, I'll pop myself in the colors here. We have salts. I'm using the term salts kind of loosely. With salts, I'm referring to sodium and chloride, magnesium, sulfates. So salt ions can be a variety of things like sodium, chloride, and magnesium. And so I'm just using the general term salts to refer to dissolved ions. Okay, so here's the thing. In chemistry, if you've had Chem 1A, you've probably learned about this acid-base equilibrium thing. And I don't totally understand it. I haven't had chemistry in like 40 years. But anyway, it's called the Le Chatelier's principle. And the Le Chatelier's principle has to do with acid equilibrium and solubility of compounds. And let me just kind of summarize this in a biology context is, if you have an acid and you put that into a neutral or basic situation, the acid dissolves in solution, right? So if we have uric acid, which is an acid, and we put that into a neutral environment, or maybe an environment that's slightly basic, the acid will actually dissolve and go into that solution. But if we put an acid in an acid, sometimes one of them will win and one will lose. And for example, uric acid in a pH 5 environment is not soluble. It's going to have to lose. So that's my, I'm not a chemist explanation for how that works here. So here we go. Uric acid solubility versus pH. If the pH is less than 7, the uric acid precipitates or turns into a solid. And if the pH is 7 or greater, the uric acid is soluble. Right? That's a really important key for how this works. And I don't think the insects do good chemistry. I don't really know, but this is how it goes. That in the pH 7 environment, the uric acid is soluble, meaning that it dissolves in the water. That's a key thing here. All right, so what happens is there are salt ion pumps, protein pumps that will actively pump salts into the Malpighian tubules. So the Malpighian tubules become hypertonic to the surrounding hemolymph, right? As a result, what happens is water and uric acid that's soluble gets sucked into the Malpighian tubules by osmosis. So the energy needed is to pump the salts in there. They make their Malpighian tubules very salty or hypertonic. And as a result, the water gets drawn in because of osmosis. And passively, the uric acid, which is soluble in the water, will get pulled in as well. Okay? All right. So all of this is then transferred to salts. All of this is then transferred to the water and uric acid. Okay, so in the hindgut at pH 5, uric acid precipitates. So it's insoluble. And we end up with a precipitate. Because it turns solid. So then there are salt ion pumps that will actively pump the salt ions back out of the hindgut. So the salts get pumped back out of the hindgut. And because the pads around the hindgut are hypertonic to the gut, the water gets pulled out along with it. Okay? So what happens basically is we're using salt pumps to draw water and uric acid when it's soluble into the Malpighian tubule. And then we're using the change in the pH. They have proton pumps that basically, kind of like your stomach does, where you pump hydrochloric acid into your stomach. They have hydrochloric acid pumping in here to make that environment acidic. But it's the change in pH that causes this uric acid to turn into a crystal and separate from the water, such that when the insect defecates, the insect releases its fecal waste, we have we have feces plus crystal uric acid. And this is called, so it's a mixture of the brown stuff is the fecal material of the food that's passed to the gut. Kind of the white little flecky crystal material is uric acid. And this is called frass. So insect feces, like cockroach feces or termite feces especially, or even if you have spiders in your house, like in the corner, sometimes you'll see like black and white droplets or black and white little solid stuff underneath where the spiders are hanging out. That's called frac because it's a mixture of feces and uric acid kind of combined together. So anyway, the cool thing about this to me is that it's using this pH to change the solubility of uric acid. So it's a combination of the mouth-picking creatures and the hindgut working together that is responsible for separating the uric acid and excreting. As a result, arthropods like scorpions and many insects can live in the driest, hottest desert environments, and they never have to really even drink water. Like scorpions get all their water they need from the prey they eat, and their metabolic water to be produced from that, so they don't have to go find drinkable water anywhere. So that's one reason why the arthropods, the insects and the scorpions and spiders, insects and chelicerates, have become like the superstars of living in the driest, hottest environments because they're able to do uric acid excretion. Okay? Sound good? Okay. All right. I think that is it for the insect physiology or arthropod physiology. So plan on a pretty, significant question on the exam that asks you to describe how mouth-picking tubes work, right? The key features are, like the crustaceans, they have no cilia, so they can't make a nephridia or use a nephridia to do this for them. So they have developed these branches off the gut tract that may or may not be at all related to the Greenland idea, who knows. But the gut tract now has become the nitrogenous waste excretion feature. And what they do is they use salt pumps to draw water and soluble uric acid out of the hemocoel. They transfer all of this into the hindgut where at a pH 5, the uric acid precipitates into a crystal and the salts and water are resorbed back into the hemocoel by salt ion pumps. And so the uric acid is solid. And probably for one thing, you should know what the term frac is, right? So that's a mixture of feces and uric acid. All right. Okay, cool. Well, I think I think that let's see here. I think we'll be able to. Trying to think here. Okay, good. So let's see what's going on here. Hang on a second. Trying to think what we move on to next. Let's go ahead and do. Trying to think here. Yeah. Okay, that sounds good. Let me switch gears here. With what I'm deciding here is I'm going to switch gears to a whole new, brand new topic, and I think what I'll do is, because I want to use the board, let's take a shorty break. Okay, I thought about changing over. I want to talk about muscles and not muscles, but I want to use the board, but I don't want to, cloud this up. So let's take like a short break. Lab today is really easy because there's no microscope use. It's all kind of big stuff. Let's take a short break. I know it's only a half an hour, like a short 10-minute break. We'll come back, and I want to jump into muscle cells and muscle physiology. And then what that'll do is it'll set us up for next week to be able to finish muscles, the nervous system, and get on our test. My fear is if I don't jump on this now, we won't have enough time for our test. So let's take a short 10-minute break. Get pictures if you need it. You probably want a picture of the test. And then we'll come back and continue until another half hour. Sounds good. Get a picture, and then I'll get the next